From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 57, recorded on June 23rd, 2020. I'm Vincent Larochelle, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey there, great to be here again, Vincent. I'm coming to you from LD Home Studios and Nursery.
Nursery? What happened there, Nels?
Yeah, some big news, some big personal news to share. My wife, Ann, and I just welcomed a daughter into the world about 2 weeks ago. Hannah Minnow LD is her name. And actually, so I am literally in her nursery right now. She hasn't been here. So we're up on the second floor in the attic of LD Home Studios, but it's a little warm, stuffy up here in this old house. So Hannah's been living on the first floor. And so the podcasting has moved to their nursery. But things are going really well. We're having a blast.
Congrats. Very good job.
Thank you.
You want to bring in our guest, Nels?
I do. So I'm really excited, Vincent, for this month's TWiEVO. It's been a while since we've been together, and we'll have to cover some of the major topics. It seems like every episode we record now, some life-changing things happen just in our communities, our society, or, for me personally this time. But so we've been, on TWiEVO, we've been talking a bunch about some work coming out of Seattle. This is by a fellow called Trevor Bedford. He's at the Fred Hutch in Seattle, and he and his crew are running this nextstrain.org. The genomic epidemiology is one way of describing it, of SARS-2. We've actually walked through his website, and some of the really covered that on TWiEVO. And so I'm really excited. So we don't have Trevor on today, but it's sort of a 1-2 punch of virus evolution coming out of the Fred Hutch. This is Jessie Bloom, another one of the incredible investigators up there, and some of his crew that just dropped a really cool preprint onto the bioarchive in the last week or so. I think it was maybe late last week. And so our three guests today, not only have they done this great work, but they're joining us, which is really fun. And so in addition to Jessie, who's here, and we'll go through with biographies in a moment, we have the two co-first authors of the work. So Allie Greeny, who I believe is an MD PhD student in Jessie's lab. She can correct me in a moment if I'm wrong. And then Tyler Starr, postdoc, and Tyler's last name fits the description, rising star in science. And so I'm really just absolutely excited for rolling up our sleeves and tackling some of this work. So the preprint is entitled Deep Mutational Scanning of SARS-CoV-2 Receptor Binding Domain Reveals Constraints on Folding and ACE2 Binding. And so before we get into the science, Vincent, why don't we introduce our guests today?
You bet. So Jessie, welcome.
Thank you. It's great to be on TWiEVO.
I know Jessie's father. I've visited him a couple of times out in Montana, actually. And I know he's a fisherman. Are you a fisherman by any chance?
I am a fisherman. I don't fish as much now that I live in Western Washington as Montana, because the fishing's a lot better in Montana. It's kind of hard to move somewhere else and fish because it's not as good, but I still do it some.
I know your dad has a license plate, Dr. Trout, right?
That's right. His license plate is Dr. Trout.
Yep, I have a picture in front of it. Well, welcome to the show. Allie Greeny, welcome to TWiEVO.
Thank you. It's great to be here.
And Tyler, you're still there because I saw your picture's gone, but you're still there, right?
Yep, I'm here.
All right, welcome. We love to start getting your histories. So let's start with you, Jessie. Now, did you grow up in Hamilton, Montana?
Yeah, I grew up in actually a town called Corvallis, Montana, which is right next to Hamilton, Montana. And I was there all through high school, and then I moved to University of Chicago for college.
Mm-hmm. So your dad tells me that you grew up with all these scientists there, including like David Baltimore, and who's the immunologist who goes out there?
Yeah, definitely. There's a lot of, because Rocky Mountain Labs is in Montana. There's always been like a very high volume of scientists going through. So it was really great as a kid growing up there. I got to meet all these scientists who would constantly be coming out to Montana
Irv Weissman, you know him, right?
That's correct, yeah.
I mean, and Stan Falco. I mean, these are guys who didn't live there, but they had summer homes because they liked to fish. So they would go, and your father said they were a big influence on you becoming a scientist. Is that true?
Yeah, I think so. Yeah? I think that's definitely true. And it was also nice for both my dad and myself growing up there because they all bought really nice properties on the Bitterroot River where we could then go fishing and duck hunting.
It's very cool. Yeah. Well, last time I was there, I went to a barbecue at your parents' house. That was really nice. Nice place. Your dad said I should spend a summer out there. That could be nice.
Oh, yeah.
So you went to University of Chicago as an undergrad, right?
Yeah, that's correct.
And you did some lab work while you were there, is that right?
Yeah, when I was an undergrad at University of Chicago, I worked with Susan Lindquist, who at the time was at University of Chicago. When I was an undergrad there, I remember meeting Nels because one of my best friends, who was another undergrad there, was in Aaron Turkowitz's lab, and I remember took me to one of these grad student happy hours, and I remember introduced Nels as a really cool guy. So, nice. and as I'll get to, Nels came back to influence my life later on. And there I worked, Susan Lindquist's lab was studying prions, which are, proteins that fold in unusual ways that can, in the English she was studying, be important for evolution. And that got me really interested in sort of the evolution of proteins.
Yeah, and of course, I remember that too, meeting at the University of Chicago and thinking, look out for this guy. He's really knows what he's doing. And so that's been proven correctly over and over again. So it's really fun to kind of think about those deep connections in science. I mean, so I think you'll soon kind of connect that thread that you brought up, Vincent, through David Baltimore's lab, but then also, when I was a grad student at Chicago and Jessie was an undergrad, and some of those overlaps, it's really fun how these collisions happen from time to time and seem to occur throughout our careers.
So where did you do your PhD?
And I moved to Caltech, and I did my PhD in chemistry, and I did my PhD with Frances Arnold, who is primarily known for protein-directed evolution. So in her lab, I was studying protein evolution, especially from an experimental standpoint, where I was interested in the idea that you could, that people, especially her lab, were mutating proteins and other enzymes and seeing what the impacts of mutations on those proteins were. And that got me super interested in concepts in evolution and protein evolution. And then when I finished that up at Caltech, I really had loved being in her lab, but her lab is very engineering focused, and a lot of people from her lab really go and tackle very applied problems in enzyme engineering, and a lot of them go into industry. And I realized I was a lot less interested in that, so I wanted to sort of study protein evolution in a context where it mattered in biology. And so I think viruses are a great example of that, because for higher organisms such as ourselves, a lot of the interesting evolution has to do with the regulation of genes, and protein evolution often isn't a major mode of sort of phenotypic and adaptive evolution. But viruses, particularly small viruses, are essentially entirely protein coding, and pretty much all of the evolutionary action is happening in the evolution of these viral proteins. So I was interested in that idea, and I remember going to talk to David Baltimore, who I knew because he was also at Caltech, and I knew because he used to go to Montana, and sort of asked him what sort of viruses would it be interesting to study this sort of evolution. And I was very interested at the time. There were a number of labs that were, for instance, using bacteriophage and other sort of viruses as sort of models for these evolutionary processes. And the advice he gave me is that, if you're interested in studying virus evolution, why don't you study a virus that people actually care about, like influenza? And that seemed like really good advice, and no one else gave me that advice. So even though his lab is, primarily known for virology from the standpoint of immunology rather than evolution, I just thought that was really good big picture advice. So I joined his lab for my postdoc and worked on virus evolution there. During that time, I went to a Keystone meeting. I met Nels, who I just started a postdoc at the Fred Hutch with Harmeet Malik. I'd never even heard of the Fred Hutch before, but Nels was talking about this cool work he was doing, and Sarah Sawyer, one of Nels' colleagues at the time, was there and talking about the really cool work she was doing. And I started to look this up, and I definitely knew about the Fred Hutch after that, and that, I think, probably played some role in me ending up out here for my faculty career.
So out of David's lab, you went up to the Hutch then?
Yeah.
You've been there ever since. So in David's lab, you did influenza, but he had not worked on influenza, right?
Yeah, so, apparently he told me he had worked on influenza, probably actually several decades before your time, Vincent. And he said Richard Franklin was the last person who worked on his lab, or was in his lab working on influenza. So, as you probably know from David's lab, one of the amazing things in his lab is that, his lab is almost more like a department because he knows so much about everything. And he's also always willing to give people enough rope to hang themselves with. So he's like, go work on influenza, sure. It was a good lesson for me because I hadn't appreciated being a graduate student in Francis's lab.
Yeah.
I was able to do a lot of experiments using tools and protocols and reagents that had already been developed by other people.
Yeah.
And so I thought, all right, science wasn't that hard. And then all of a sudden I was trying to figure out how to work with influenza with no one around who knew how to do it and no knowledge myself. And it was a good learning experience.
So we should connect the loop here, Vincent. So you were also in David Baltimore's lab. You don't have to say exactly how many years.
I can say I'm an old guy. It's no problem. I'm the oldest guy on this show. It's fine. I was in his lab from 79 to 82 as a postdoc, but he was at MIT at the time. But nevertheless, you're right. I mean, so I was in the polio group. He had a retrovirus group, and then he had an immunology group, which came out of the finding of one of these retroviruses, and the way it interacted with it. So it was just amazing. And this is a guy who would go away for a couple of weeks and come back and sit down, and he just picked up where you left off. It was really remarkable. It's really something. Now, Jessie, I remember you came here and you gave a talk, and Ian Lipkin had invited you. And I remember you talking about influenza virus, and you had stuck GFP into it so you could do rapid measurement of reproduction. And I remember you saying, plaque assays are too much work, so you developed this, which is, for me, plaque assay is my favorite assay in the world. So I said, what is this guy talking about?
I know. And it's funny, my lab still now almost doesn't do plaque. We do a lot of PCID50s, but flu's not the easiest virus to plaque. But we now have a postdoc, Bernadette, in our group who came from Urban Fodor's group, who has a really good training in flu virology, and now she's doing plaque assays. So now our lab is
so right here in my office, I have a wall of plaque assays, 1,600 6-well plates of poliovirus plaque assays. They're all stained, and it was one experiment. It's like my masterpiece. Yeah, Nels, you've seen it. I have a picture.
Yeah, we've got a nice photograph in front of it.
So if we ever travel again, and any of you come by, you have to get your picture taken in front of it.
Definitely.
Allie, where are you from?
So I'm from Castro Valley, California, which is in the Bay Area. So I grew up there, and I went to college at Berkeley. And when I was in college, I was in Nicole King's lab studying choanoflagellates. And I was really lucky there to be mentored by great science mentors. Nicole was a great mentor, and I was lucky to be working with a graduate student at the time, Tara Levin. And so I think working with her really inspired me to continue a career in science. And she is now, or she was a postdoc in Harmeet Malik's lab at the Hutch as well. But I think she's off starting her own lab now at Pittsburgh.
Yep, that's exactly right, Allie. So again, a small world in science, right? So,
yeah.
Sure.
And just to circle back for a second. So Jessie mentioned Sarah Sawyer, our great colleague. If you haven't heard about some of her work, or you can hear from her directly actually on I think it's TWiEVO number 2. It's in the early first handful of the episodes we recorded and had her on, as well as Kartik Chandran. That was a really fun episode. And then Nicole King was on TWiEVO as well. And so we can hear Allie's mentor there. And lots of connections here. Oh, and then Tara Levin, who was, as you're saying, postdoc with Harmeet, and is now opening, I think she's there actually, and has officially opened her lab in Pittsburgh. Keep an eye on the Levin lab. And in fact, one of the grad students from my crew, Della Fixon, is just interviewing for a postdoc in Tara's lab now. So it's all kind of intermeshed here.
And then there's one more connection, Nels. So, and Allie, you'll recognize this. So Nels and I and Rich Condit were in Germany for a giant virus meeting. And who was the keynote? What was her name, Nels? I forgot.
I'm totally blanking on it at the moment, but
anyway,
I know.
She had isolated a choanovirus, a giant choanovirus. So he's talking about Nicole, and Nicole was, and I think they were on field trips or something together, right?
Well, that's exactly right. So that was where were you on that trip, Allie, by chance? Maybe
the Mono Lake trip, or that's the only trip I've been on.
I think it was a different one in on the Caribbean island, Curacao.
Not lucky enough to go there. No.
You have to be like kind of advanced grad student or postdoc for that access. Yeah. But yeah, exactly. I think they, Nicole, and then so if you listen to the TWiEVO, the video TWiEVO, I guess, or no, sorry, the TWiV, but it was sort of a hybrid episode. We covered that work as well.
Alexandra Warden.
That's it.
You must know that name, Allie, right? Alexandra Warden.
I think I actually heard about this work on TWiV or TWiEVO, whichever episode you guys did.
Yeah, that's right. So she found this giant virus in the ocean. Turns out the host she was pulling single cells out of the ocean with a fax sorter on a boat. She told us she spent her whole startup money on this fax sorter. and it turned out to have a giant virus in it. And so now she said she got Nicole excited about looking for more viruses. Anyway, so then after that, you went to Jessie's lab, right?
Yeah, I did a 2-year stint as a postbac at the NIH in Bethesda, and then came to UW for medical school for the MD PhD program. And so I started here in 2016 and then joined Jessie's lab in 2018.
So Allie, you did your first couple of years of medical school before getting back to the research, is that right? Yeah. And how did you pick Jessie's lab? I mean, I know he's literally listening to us, but
yeah. I think so. I met with Jessie when I came for second visit, and was really excited about his work, and everyone who I talked to is really excited about the lab and the environment. And I did my first rotation in Harmeet Malik's lab, actually, and then my second rotation in Jessie's lab. And I think a big part of kind of something that Jessie alluded to earlier is that in his lab, it's a really great training environment for graduate students, I think, because Jessie is very organized and has set up all these really organized systems that make it possible to enter into the lab and kind of get started on a project relatively quickly, which is always important for MD PhD students on a relatively rapid timeline. And so, for example, one thing that was, this is just a small thing, but it was actually huge, is that Jessie has this very organized plasmid log of all, I think now we're approaching 3,000 plasmids that have been cloned in the lab. And you can look in the log and find any one of them and get a small aliquot and get started right away on anything that you're working on. So
I tried doing that in my lab, and as soon as people came in, they wouldn't do it. So the book has 150 entries, which are all mine. And then after that, nobody else put anything into it. So congratulations on being able to do.
Yeah, right. Organization's not my strong suit either, but
I'm not totally unorganized in the lab anyway. And myself, I couldn't organize. I couldn't get other people to do it. So it's a skill, getting other people to buy into what you think is a good way. That's really important. You have to be convincing. So he must be convincing, I guess.
Yes, I think that's one of Jessie's skills as a mentor. He can really take. He can work with a variety of people who have very different personalities and tailor his own mentoring approach to them in such a way that everyone can work together cohesively, and he can still get the plasmid log that he wants in the end. Yeah.
Hey, Allie, can I ask, it's kind of early days, but what's your thoughts on sort of that balance between medicine and research science? How do you think about that?
Yeah, it's like the big existential crisis that every MD PhD faces. I think I am super interested in, continuing a career in research, and I suppose the traditional MD PhD path that you kind of hear about or is kind of held up as the typical route or the most ideal route of spending the majority of your time doing research and running a lab, and then some minority of your time engaged in clinical activities that ideally would relate to your research in some way. So And I think a lot of the research that I've been able to do in Jessie's lab, I can see where there would be very obvious clinical things that I could, clinical things that I could do that would be, related to some really exciting research.
Yeah, very cool. And probably already are in touch, but I would certainly hold up Adam Jabal in the neighborhood there at Fred Hutch. Mm-hmm. So MD, but very much research focused. And he was one of my key mentors as I learned some pox virology late in my postdoc training, and still one of both a great mentor and close friend.
Yeah, Adam has already offered for me to come and shadow him on the ID wards. So
awesome. Good idea.
Tyler, where are you from?
So I grew up in Nels' neck of the woods in Minnesota, Hopkins, Minnesota. I forget what town Nels is from, but
South
cities.
Yeah,
oh, that's right. Yeah. So yep, I grew up in Minnesota, but then I came out west for undergraduate, where I went to a small liberal arts college called Willamette University, which is in Salem, Oregon. And there I started research with Chris Smith, who worked on the Joshua trees of the Southwest Mojave Desert. So my first research
of the desert and doing transects in the field on Joshua trees. But, as I went through college, I got really into the molecular world and the world of proteins, which I know you all talk a lot about, sort of these fusions of fields. And so that's what led me to my PhD, which I did at University of Chicago.
So, and Tyler, so that remind me, so you did your PhD with Joe Thornton. Mm-hmm. And so this was after he had moved his lab completely from Oregon, or did you have a stint in Eugene as part of your PhD training as well?
So I started my PhD in University of Chicago with him there. I did do an NSF research experience for undergraduate, REU summer in Joe's lab at University of Oregon before my senior year of college. So that's where I got to know his lab and got to spend some time with that Eugene component. But as I was applying for graduate schools, that's when he was moving out to Chicago.
Oh, okay. And so were you actually thinking about his lab when you ended up in Chicago, or how did that kind of connection come to be?
Yeah, I actually tried to stay on the West Coast for grad school. I think I applied to all West Coast grad schools, except then Joe told me, if you want to do your PhD with me, I'm probably moving. So I threw one Eastern school in the mix, and lo and behold, ended up back in the Midwest. But yeah, actually kind of the final decision, funnily enough, was basically down to UW with the probable intent of joining Jessie's lab for grad school versus Joe. I know everyone always says your interests are going to change and you'll probably end up wanting to work on something else in the future anyway. But then lo and behold, for postdoc, I ended up in Jessie's lab anyway.
And I think I actually worked out great. I actually remember it was my first year as an assistant professor. I think when Tyler was going there, I was very disappointed he didn't come here. But as awesome as I'm sure Tyler would have done as a grad student if he joined my lab, it's even better to have him go and do like 5 years of amazing training in Joe Thornton's lab, and then have an even wiser version of Tyler and join the lab as a postdoc. So
that's cool.
I'm now glad you didn't come here for grad school, Tyler.
That's interesting. Second chance. Yeah.
Well, and I have to say, it was almost like, I would say in the field of evolutionary genetics, lots of folks were familiar with what Tyler was up to in Joe Thornton's lab at Chicago. And I'll let you fill in some of those details, but it was almost like a secret postdoc sweepstakes. Where is Tyler going to end up? And so we were all a little bit jealous, but also happy when he landed in Jessie's lab. What a great connection.
I remember when I rotated in the lab, like the next month was when Tyler came for his postdoc interview. And so then I was gossiping with all the grad students, and everyone was like, I don't think Tyler's going to come. He's too cool for us. But everyone was very happy and relieved when he decided to come.
As was I.
Great. All right, Nels, there you go.
Yeah. So great to have this crew with us. And I should say, we've already kind of covered a little bit of the ground here. So usually thinking about influenza evolution, and I would say really a unique combination of evolutionary virology and biochemistry, and that'll be in display today. So for this preprint, but I guess, to begin with, obviously we're making a massive left turn or right turn here, some kind of turn away from influenza into SARS-CoV-2. And so Before we get into the work here, maybe any of the three of you, if you want to tell us how did you get onto this, or how did you make this transition from influenza into SARS-2? What was it that motivated the study?
I think Tyler might be the best one to answer that question because he sort of was the one who got the ball rolling. On it.
Yeah, well, at least from like the experimental side, I'd spent my first year of my postdoc setting up this assay system, which we ended up applying here kind of as a, rabbit jump over my original project. And so I'd kind of optimized this experimental system and uncovered a lot of the problems that I'd been running into in this other system. And was kind of getting ready to kind of do the final push on a project there, right when SARS-CoV-2 started really lighting up. And so I think kind of the proximal origin of all of this was obviously, as the more virally attuned of us, Jessie was, getting really, really into all the SARS-CoV-2 research and evolution stuff. And I think he just saw a paper or something like that in which this receptor binding domain, this RBD that we're going to talk a lot about, was produced in yeast or something like that. And this yeast system is what I was working on. And so kind of in the hallway one day, he just goes, Tyler, like you can express RBD in yeast. Like, do you think you can just throw this into your yeast display system? And I was kind of laughing in hindsight. My exact words were like, oh yeah, it'd probably work, but I don't want to get too distracted. And so then he started a Slack channel, which is our lab communication page, on this project that day. And we just started ordering genes and, kind of started rolling from there, I guess.
What month are we talking about? January or February?
It was end of February.
Okay. What were you doing with the yeast expression before? Was it an influenza project or something else?
It was an HIV antibodies project.
And then, so as co-first authors, maybe Allie, maybe help us understand, how did you get into the picture here? Or how did you connect the division of labor?
Yeah. So the way that I got involved in the project was, during what I felt was a very stressful time when the pandemic was, it was starting to become clear, like how serious the pandemic was locally. And so the Hutch was transitioning to pretty much, like one person per lab. And so everyone, was kind of like trying to figure out how they were going to continue their work. And I was standing by the thermocyclers talking to Jessie and was like, oh, Jessie, like, do you think Tyler needs some help with this project? and Tyler, and so Jessie was like, yeah, I think actually that would be good. But like, you can't use this as an excuse to work on your neuraminidase stuff. If you're going to work on this, like, this needs to be your full-time project for now. And so then I got pulled in that way.
That's great. So, and Jessie, maybe what's going on in the lab overall now? Have you shifted completely to SARS-2, or what's the balance of things? How are you thinking about that?
I mean, probably like a lot of virology labs, we started to pay a lot of attention to SARS coronavirus 2, and there were, a number of people who were doing sort of various things related to SARS coronavirus 2, probably 4 or 5 people, and for a while that was almost all the research that was going on in the lab, because there's still quite a bit of, with good reason, limitation on, how many people can be in lab at any given time here at the Hutch. And particularly when it was very dialed back on the number of people that were allowed in, we were just prioritizing the type of people who were doing work related to SARS coronavirus 2. So in addition to Tyler and Allie's project, which I'll tell you a lot about, Kate, who's actually another grad student in the lab who's on this paper, set up, and she didn't invent this, but she did a very nice job of getting it work nicely, like a lentiviral pseudotyping system for the coronavirus spike protein, which is actually a little bit in this paper because it was used to validate a few mutations. And then we've also been doing a little bit of serology work, but definitely our lab is not primarily, the majority of people are still probably not doing SARS coronavirus 2 work, but I think, we were trying to find things where we could contribute for a little while. And then an important, obviously it's not on this podcast, but we were very fortunate that David Wiesler, who's a co-author on this paper, who's an assistant professor in UW biochemistry, and he's really perhaps the world's expert on coronavirus spikes. And importantly, he was working on this before SARS coronavirus 2. He was using cryo-EM to really understand how these spike proteins work, and their mechanism is in some ways analogous to, let's say, how influenza, hemagglutinin, or HIV envelope work, but they're much bigger and much more complex proteins, and his lab had been studying those for a number of years. And so some of the work we were able to do, like what Tyler and Allie have done, we've benefited a lot from, just reading David Wiesler's reviews, but also being able to talk to and collaborate with someone who, has been thinking about coronaviruses for more than 5 months, which has been very useful.
No, that's right. It really is a kind of an interesting balance with a lot of labs. So my lab included, nothing to kind of this level, but one of the postdocs in my group, Stephen Goldstein, who got his PhD in coronavirus biology from Susan Weiss's lab. It's sort of a natural extension, but that's been an interesting balance that we've certainly discussed a lot on TWiEVO over the last few months, is how labs sort of adjust, and sort of that balance, or that notion of, in some sense, staying in your lane, but also taking advantage of opportunities. I think this is a great example of a study where it really does make sense to sort of reach and extend, given some of those local connections and the expertise here. So we're going to dive into deep mutational scanning in a moment, but maybe let's actually set up the biology first. So the SARS-2 receptor binding domain of the spike protein, what is it? Why does binding matter? What is it binding to? And maybe Allie and Tyler, you could help us out here to just sort of set the table for the biology that you were examining in the preprint.
Yeah, I can start off. So as Jessie was explaining, this surface spike protein of the SARS coronavirus 2 is kind of how it sees the world. It's external protein on the viral surface. And it uses this spike protein to basically bind to host cells and then enter those cells to release its genetic material and do the rest of the stuff that it does. And in particular, it's this small domain of the spike protein called the receptor binding domain, or the RBD, that actually mediates that binding to the ACE2 receptor on the host cell surface. And so it's that high affinity interaction between the RBD and ACE2 that allows SARS-CoV-2 to, jump from one host to another and enter cells and mediate that infection.
And remind us, either Tyler or Allie, how big is the RBD? How many amino acids are we talking about?
Yeah, so it's only about 200 amino acids, I think. The particular construct that we're working with, I think, was 201 something.
And you're mutating across that entire stretch of 200 amino acids or so? Yeah.
Right. Mm-hmm.
And we had already known that if you produce just RBD on its own, it will fold in a way that can bind to the receptor ACE2, correct?
Right.
Yep.
Mm-hmm.
So especially from all the SARS-CoV-1 literature, there was a lot of structural work on these RBDs, as well as a lot of actual evolutionary work, kind of identifying what amino acid substitutions in this RBD allowed it to gain human ACE2 affinity in that original SARS-CoV coronavirus outbreak. And then there was pretty much immediate interest in the RBD of SARS-CoV-2 when its sequence first came online. And actually, that was kind of one of the things that was sort of interesting, was that the RBD in a lot of these positions that were known to contact ACE2, the amino acids it used were different than what SARS-CoV-1, as we call it, used. And so it was immediately clear that this was, obviously evolutionarily related, but it's sort of a distant cousin of that original SARS-CoV. And its RBD is binding ACE2 in some sort of slightly different way, sort of at the atomic level with the individual amino acid residues.
So
I think
Go ahead.
I think one other kind of piece of just background literature that was, sort of to just develop the process of how we thought about this project that was really important is very early in this pandemic, there was a very nice paper. I can't remember if either Vincent, you've covered it on TWiEVO, or it's been covered somewhere else, by Michael Letko and Vincent Munster.
Yes.
Actually Rocky Mountain Labs. And in that paper, they were sort of exploring, I think they'd done most of the project before SARS coronavirus 2 emerged. They were exploring which receptors different coronaviruses could bind to. And one of the key ideas in that paper is, although it's not complete, at least to some degree, the receptor binding domain is modular. And they were performing that study by taking receptor binding domains from different beta coronaviruses and transplanting them on just one beta coronavirus backbone and seeing which cells it could infect. And so that was both a very important paper sort of biologically, because it was the first, or one of the first, to show that this virus bound to ACE2. But I think for a lot of work, including our work, it was also very important because they kind of had this nice idea of, at least to some degree, that you could think about and study this in a little bit of a modular way.
Right. Yeah. Vincent had sent me that paper very early on, and it was like the first SARS-CoV-2 paper I saw, and that was very cool. Yeah. So Tyler, how do you produce a protein on the surface of yeast? So this RBD on its own would not necessarily do that. Do you have to stick it onto something?
Yeah, you just make it as a sort of genetic fusion with a natural yeast cell surface protein.
Okay.
And so then you end up with this, something like 10,000 or 100,000 copies of whatever protein you put there. In this case, the RBD, just tethered to the yeast cell surface. And then it's really nice because once it's there, you can kind of do any sort of fluorescent-based measurement of kind of RBD, biochemical functions.
How many yeast cell surface proteins are there normally? Not how many different ones?
Oh, I'm not sure.
I mean, are there thousands or just a few? Does anybody know?
Yeah, that's a good
I
question.
have no idea. I didn't know there were any, actually.
I'm sure there's many. At least
many.
I'm guessing it's less than thousands because I think the whole yeast proteome is only 3,000 or so.
Yeah.
3,000 or 4,000 protein coding genes.
I mean, there aren't that many in mammalian cells, actually. It's just in the thousands, I think. So, if you want to look for a virus receptor, you could just sort through all the known membrane-bound proteins. It's not a lot. So, I just was not even aware that you could do this. I view yeast as covered with a fingernail, basically.
Yeah, no, this yeast display system has a pretty rich history. For 20 or 30 years. It's been used a lot, especially in like antibody engineering. That's a big field where yeast display has been used a lot to, fish out antibodies from repertoires or kind of engineer your own high affinity antibodies. But why we were getting into it was just a few years ago, it was published as a sort of link to one of these so-called deep mutational scanning assays in a sort of high throughput format where you can use yeast display of a protein of interest, but you can actually generate libraries of many different variants of your protein of interest and do these titration curves. So these careful, like pseudo biochemical measurements of, binding strength. But you could kind of do it all in one pot reaction and get, thousands and thousands of binding measurements of biochemical value sort of all at once.
So when you first put the RBD gene into yeast as your fusion, I guess you first looked to see if it was on the surface, right?
Yep, that's exactly right. So, within a week or two after Jessie said, hey, we should see if RBD works in yeast, I'd ordered a gene from IDT, cloned it into this yeast display plasmid, and then just went down to flow cytometry and could see that, the RBD is there on the yeast cell surface and it seems to be folded, and it's functional there. We can incubate with soluble ACE2 and we can see that these yeast like sop up that ACE2 out of solution. So
that must have been exciting, right? Yeah,
it was quite exciting.
Fast.
So, and then, exactly. And then, this is not much time from, when the pandemic started to the preprint emerging here. But so, and Vincent noted the, potential complication of what else is on the yeast surface. And certainly that will be consistent throughout. But there's also another point here, right? Is that the spike protein or the receptor binding domain becomes glycosylated. What do you how do you think about that, Allie? Or how does that kind of connect into the work here?
Yeah, so yeast do glycosylate proteins as well, but the glycosylations, I believe, are more mannose rich, so they're different.
So, as soon as this gets published, people are going to write you and say, can I use this to prevent COVID disease? It's inevitable, right? They're going to say,
You already had.
Okay. Yeah.
No, we already had one Twitter thread saying that our study somehow supports the lab origin of the virus. I have no idea how the heck you get that.
Oh, yeah, sure. Sure.
Yeah.
Oh, right.
And I do think, Vincent, to go to your question, although I'm not that up to speed on the SARS coronavirus 2 vaccines, I think there's at least some vaccine candidates that are, or certainly there are some vaccine candidates that are solely RBD protein. And I think at least some of them are produced in yeast.
Mm-hmm.
I can't remember exactly which vaccine that is, but I think there's at least one vaccine. I think it's maybe not Saccharomyces cerevisiae, but I think there's at least one vaccine that's produced in some other yeast strain, which is the RBD.
No, you're absolutely right. There are some RBD only vaccines in I'm not sure that that's a particularly great idea, but we'll see. They're all going to be tested at some level, right? So Tyler, as soon as you found that it worked, then you decided to go on and introduce all the changes into it?
Yep. So that's when, as soon as we had that first result, basically we were ordering our primer library to introduce all these mutations. And that was right when stuff was getting close to shutting down. So that's right when Allie jumped on board, and she has a lot of experience in making these mutant libraries. So she knocked that out of the park.
How did you do that, Allie?
Yeah, so it's a PCR-based mutagenesis method. So we order primers where kind of in the middle of the primer is an NNN, or in this case, NNS, or you could also use NNK. And those are just degenerate nucleotide codons to mutagenize every codon kind of one by one as you go across your gene. And so for every codon in the gene, you have a different primer. And so we ordered these, and you can actually order them now, which is different from the last time that I made a library, in pools, like pre-pooled. So they just come in a tube, and you take your primers out of the tube and you mutagenize your gene, in bulk, and you do your cloning in bulk. And so at the end, you get a plasmid library where every plasmid, where your plasmid library represents every possible codon mutation to your gene of interest. And then we transform that library into yeast. And then it's a low copy plasmid that resolves down, basically has like a centromere on it. So it resolves down to a single copy per yeast cell. And now you have a whole pool of yeast cells where every cell expresses one unique mutant protein on its surface. And so you have that nice genotype-phenotype link, and you can perform your assays on that pool of cells, kind of maintaining that link between the phenotype on the cell and the plasmid that's carried inside the cell.
So in this process, what you're describing is the deep mutational scanning approach. And so how do you connect that or think about that in terms of like sort of the natural evolutionary process? How does it sort of correlate to that or sort of approximate that?
Yeah. So, viruses are rapidly or have a higher mutation rate than, say, humans, although coronaviruses do have a lower mutation rate because they do have proofreading. But kind of the advantage of the deep mutational scan is that we can sample, all mutations that have been sampled in nature, but we can also sample those that have not yet been sampled in nature. And we can even sample those that are more than one nucleotide mutation away from, whatever the wild type sequence is. So we can sample those 2 or 3 mutations in a single codon, which are really unlikely to happen in nature, but we can kind of accelerate the process a bit in the lab that way and get all of those measurements at one time.
And you don't have constraints of virus replication because you just have a protein on the yeast surface, right? So you could look at every change, and including those which would never make it into virus, right?
Yeah. Mm-hmm.
So did you get every amino acid at every position in the RBD?
I think we missed a small number. I think
15.
15, yeah. Yeah.
But you didn't get every possible combination, right?
No.
That would be huge.
Yeah. Yes. Yeah. So the way that the mutagenesis approach works is that we do get, as you're alluding to, some random subsampling of multiple mutants. And we kind of restricted our library to only about 100,000 to 150,000 unique variants per library. So we have, some kind of random combinations of those.
Got it.
And just to add one more thing to sort of go back to Nels' sort of conceptual question, Allie sort of explained this, but just to elaborate more for people who aren't used to thinking about this, the difference between deep mutational scanning and evolution is evolution is a process that involves mutation and selection, but it also involves a lot of other forces. And so certainly as, for example, on TWiEVO, I feel like Vincent and his co-hosts are constantly reminding people is that when you look at the natural evolution of a virus or most things, it doesn't mean that every mutation you see is somehow making the thing better, or in the case of a virus, more transmissible. In fact, most of them aren't. There's a lot of random forces going on in evolution, things are drifting around, mutations are appearing randomly. And that's a very important part of what evolution does. What a deep mutational scanning experiment is tries to take out all of that random thing and just say, what is the functional effect of the mutation? So in a deep mutational scanning experiment, you make all the mutations once, and then you perform a single selection step to see what happens to them. And so in that case, what happens to a mutation, like whether it's enriched or not enriched or something like that, really just depends on how it affects the phenotype that you care about. And so this doesn't tell you how evolution will proceed, because real evolution has all of these random forces, but it's in some ways more interpretable data because you're looking at the effects of mutations without all of these random forces laid on top. And then a second important point that I think Vincent alluded to is in these experiments, you're sort of measuring the effects of mutations on something you can measure in the lab, which we'll describe what that is later. But we're not measuring what natural selection would measure. Like a real coronavirus is way more complicated than the receptor binding domain. And a real coronavirus has to do a lot more than bind to ACE2. So we're measuring how mutations affect a phenotype, which is important for evolution, but it's just one small fraction of the many things that are important for evolution.
Yeah, there's a great sentence about that in the discussion, which I really liked. I highlighted because many people forget this, that, what you're measuring in the lab is the lab. It's not nature. But I really appreciate that you made that distinction, and not everyone does.
Yeah,
it's really good.
It's a really important point. So then, you quickly made this great diverse library of clones. And then now I'm kind of paging ahead here to figure 3 of the preprint. And there's this really cool map, phenotype to sequence map. Am I getting that right? Or to binding map. Sorry, you'll have to help me out here. But it's worth looking at this because this is like all of this work, right? Just sort of visualized right before your eyes. So I'm looking at all of these squares that some are blue, some are red, and they're different contact zones. Help us decode this. What did you find when you sort of executed your experiment?
Yeah, so that is our data right there. Those very complex, we call them heat maps, illustrate the effect of every single one of these amino acid mutations across the entire RBD on sort of the 2 phenotypes that we measured. One of which is sort of the kind of a correlate of the stability of the RBD. So how well folded is it versus being sort of an unfolded, non-functional mess of a protein. And then beyond that, we have the effects of all these mutations on ACE2 binding affinity. So how tightly does it bind to this ACE2 receptor? And so basically the first week of data analysis just consisted of staring at these heat maps in excruciating detail and kind of just pulling out patterns of differences in the red versus the blue of what binds, what mutation affects binding or what affects expression, and how does that map to the structure and things like that. And so I think one important thing we try to do with this paper is present these data in a lot of different ways so that other people who have super diverse uses for this data can kind of do their own staring. And so I think there were 2 really important sort of interactive visualization tools that we tried to provide with this paper. And so these were spearheaded by Sarah Hilton, a grad student in Jessie's lab. And one of them is to take these heat maps and make them in a sort of interactive sense so that you can move your mouse around and read out the values on these heat maps, and also subset positions that are known to contact ACE2 and things like that. So anyone can find any mutation they care about, whether they're trying to engineer RBDs for vaccines or whether they see some naturally occurring mutation within the RBD, they can go to these maps and read out these biochemical phenotypes. And then the other thing we did was this sort of structure-based visualization where you can map these mutation effects to the known 3-dimensional structure of the RBD bound to ACE2. And kind of from that type of view, you can learn a lot about sort of the biochemistry of this domain and how mutational effects map to that structure and impact its function.
I noticed when you mouse over, so you get information about each amino acid change, what it does to ACE binding. So red is knocking it down, right? Mm-hmm. Blue is neutral or better. But also you have in this, what pops up are the amino acids in that position in SARS-CoV-1, RATG13, the closest bat SARS-like isolate, the pangolin isolate too. So you can immediately tell, because those are the three probably most relevant for the evolution of this thing.
Yeah, exactly. I think, we wanted to do a lot of different things with these data and make them useful to a lot of virology things, but I think all three of us always still come from an evolution background. So no matter what we were talking about, we were always thinking about how these things map to the phylogeny and the evolution of these viruses kind of in our background.
And if you look at this map, you get two red areas, basically, right? Two clusters of red. Mm-hmm. And what do you think that means?
As probably the person who's maybe spent the most time trying to trace that down, mainly you kind of see two regions of the protein where mutations tend to reduce its function. And they what they map down to are essentially the folded core of the protein. So if you mutate these buried residues at the very center of the protein, even though they're far away from the ACE2 interface, they cause the protein just to fall apart. And so it can't function. And so that's a large part of the constraint that we map. But then, of course, there are these amino acids where the side chains are making crucial contacts to ACE2. And if you happen to mutate them at some of these positions, you'll lose that affinity. But that actually does kind of relate to something that I thought was more so surprising, what we saw, which is that a lot of mutations at these interface residues were actually capable of improving the affinity for ACE2, which is not something you always see in these demutational scans, such a large number of sort of biochemically improving mutations.
So in here, as we're looking at the kind of the graph or the visualization of the affinity, either the expression at the top two or the affinity at the bottom two, you can see all of the different amino acids for kind of as it relates to each position. Mm-hmm. These are all non-synonymous changes, meaning the amino acid has changed. So I think you mentioned in the preprint that you've also obviously, given the degeneracy of the genetic code, you end up making a lot of synonymous mutations, meaning the amino acid stays the same. And what did you observe in terms of either expression or binding for those mutations that don't change the amino acid?
Yeah, so kind of as you would expect, or in some ways this is a good validation of our approach, that these synonymous changes do not have any effect on expression or binding, and the distribution of the fitness effects for the synonymous mutants look kind of just like the wild type unmutated components of the library as well.
Yeah, and I think I like that point, or I think it's an important sort of validation, and because we think about this a lot in evolutionary genetics, is sort of what's the influence of mutations, and we use often the signatures of the non-synonymous mutations to try to kind of highlight where we think evolution is happening in interesting ways, where adaptive changes might be happening, and it's a major assumption that only the non-synonymous changes will have these sort of massive effects, but this sort of, at least supports that to some degree, that the synonymous ones there might be a small distribution of effects, but it's much more limited. And so one of the ways in my lab, we describe this as sort of like rearranging the deck furniture on the Titanic, right? It's like, that's nice, or it might change the virus a little bit, but if you hit an iceberg evolutionarily, a big adaptive change where you change an amino acid, that is probably going to sort of drive the evolutionary signals. And it seems like that's what you're seeing.
And I would definitely agree with what you're saying there, Nels. And I think, for the evolution of this virus, frequently the non-synonymous or the amino acid changing mutations will have the biggest effect. But I do think it's important not to over-extrapolate our data in that sense, because we are looking at synonymous mutations in the yeast-expressed system, where almost certainly the synonymous mutations are not doing much. And I think even in the virus, amino acid mutations will tend to be more important than synonymous mutations. And I actually am not all that knowledgeable about this part of SARS coronavirus too, but almost all viruses, because their genomes are packaged in, some physical way, and because there's a lot of regulation of how exactly transcription happens, it's quite possible that some of the synonymous mutations might have effects on the virus itself, unrelated to changing the protein sequence. And we wouldn't find that in our assay because we don't have any of those things going on.
Most certainly. That's how recoding genomes makes attenuated viruses, right? And that's one of the vaccines being tested, actually. Change the RNA, not the protein. Messes up the virulence. Yeah, for sure.
So I'm seeing a lot of red here, not a whole lot of blue in terms of the higher affinity. So what are kind of the main take-homes from all of this phenotype sequence mapping?
Yeah, I think that's a common theme of deep mutational scanning, is that you definitely pick up a lot more constraint from the sense of many mutations break a protein. That's kind of the intuitive thing there. But I think we did quantify essentially how many of these mutations are within the window of affinity that we at least know from our N=2 coronaviruses that can infect humans. The affinities of SARS-CoV-1 and SARS-CoV-2. And we do see that almost half of the amino acid mutations we tested are kind of within that affinity window, which actually is we think a somewhat large window of kind of mutational accessibility. And it does suggest that there are perhaps a lot of mutations which, at least for this phenotype of ACE2 binding, are kind of within an allowed window. And it does suggest, which is also reflected in the evolutionary record of substitutions in this domain, the fact that there can be a lot of kind of change in this domain. It's not a highly conserved domain from the sequence perspective, even though it still binds ACE2 in these two viruses. There's a lot of amino acid changes, and our maps kind of show that that's consistent with kind of the general mutational tolerance of this function.
I think it's really interesting that you have, what do you have, a 10-fold difference in affinity between SARS-1 and 2 binding? Is that the number?
A bit less than 10, about, I think sometimes it's maybe seen to be as much 10, sometimes maybe like 5-fold.
But I like that because, as you say, they both can infect people. So that's at least the minimal range that you have to work with, right?
Mm-hmm. Exactly. And
this high affinity binding is highly unusual. I guess the pangolin is the other one with the high affinity RBD. But that's of course the big question: how that was selected for, and where in nature, right? Where is the human ACE-like receptor that selected that?
Yep.
Yeah, and we mentioned might have been a couple episodes ago that, so if you look at on the other side of the coin, right, what's going on with the receptor, the ACE2, in the potential hosts. And so if you look among primates, our closest relative, it's actually, there's not a lot of evolutionary signal there. Mm-hmm. However, among bats, there is a lot of evolution happening in ACE2. And so that could be another maybe way of approaching this, or get some clues, is to start to put together the close relatives of pangolins or other lineages to see if there are cases where ACE2 is evolving in a way that's sort of similar to bats, with this sort of rapid and recurrent kind of signatures that we talk about with adaptive evolution. I don't know if anyone's done that.
Yeah. So my sense from what I've been reading is that it does seem like the ability to bind ACE2 does just emerge even within these bat isolates. So however, this virus is evolving within sort of these bat species seems to kind of happen where human ACE2 binding can kind of just come along for the ride. And I think, like you're saying, that does really stem from these really complex evolutionary pressures that these viruses are under in bats. And so I think this first started with a paper by Sarah Sawyer, who we were talking about earlier, after SARS-CoV-1, in which she noticed that ACE2 is under really elevated rates of positive selection in bats. Or there's a more recent preprint from Zhengli Shi, where she sequenced the ACE2 molecules from like 40 different individuals of this one bat species, which harbors a lot of these viruses. And she uncovered 8 different like alleles of this ACE2 protein that differ at all of these coronavirus contact positions. Mm. And so this evolutionary arms race going on here is something wild. And we're kind of just the accidental spillover hosts, I think, in a lot of these instances.
So what is it? 5 or 6 amino acid changes in the RBD distinguish the RATG from SARS-CoV-2? Is that the right number? The ones that matter. The ones that matter for affinity. 5 or 6
contact positions. Yeah.
So could those arise, if those arose in bats, just spontaneously, right? Would there be a downside? Would the virus have a fitness cost in bats? Do we know that?
We definitely don't know that. I don't know if anyone wants to speculate.
Because, you could imagine that they would arise randomly with no fitness cost in bats, and then you encounter a human and boom. So it doesn't even have to be selected for in some animal, right?
Yeah, no, I totally agree with what Vincent's sort of articulating is this concept, which I think still, and especially when I was in my PhD, was very influential in sort of theoretical evolution, which is the concept of sort of diffusing along a neutral network, helping adaptive evolution. So the idea is there might be a big space of RBD sequences that can bind to the bat receptor, and some small fraction of them can also bind to human ACE2. And if viruses are just kind of randomly diffusing around in this network that's all neutral when you're in the bat, sometimes it will get over to a place where it can also bind to the human one. I mean, we don't know if that's true, but I think that's kind of what you're suggesting.
Yeah,
certainly,
exactly right.
It could well be true.
Yeah, put much better than I did, though.
So then, just looking at the clock here, I could talk for hours about this, but we should probably keep moving here. So, in the discussion, so looking at that constraint in particular, you make some connections to possible vaccine development. Maybe help walk us through some of the ideas about how applying some of the results that you got from this study could be useful moving forward.
I think that from this study, we can kind of get a sense of the general constraint on the protein. And, kind of if you look at, I think it's figure 7, Tyler maps some known antibody epitopes onto the structure with our measured mutational tolerance. And you can see that a lot of these, or a good number of these epitopes do have some, varying tolerance, and that, perhaps like targeting a more conserved or less tolerant to mutation region in the RBM might, make for a better vaccine than, if all the immune response is perhaps targeted towards a region that has quite a bit of leeway to, to mutate and escape from that antibody while still maintaining its function in binding to ACE2. And so some possible ways that you could imagine kind of leveraging this could be like resurfacing the RBD to kind of mask epitopes that you don't want the immune response to be targeted towards, things like that.
And I think that idea of kind of mutational escape from antibodies is underscored by a paper we reference in our preprint that came out like the day or two before we finalized it, but was extremely important. We thought this is a paper in Science from a team at Regeneron that has some of these monoclonal antibodies that target the RBD. And if they just passage the virus in the presence of these antibodies, mutations that escape neutralization by these antibodies fix. And when we look at our data, we can see that even though these antibodies do bind regions that have mutational constraint, they also have some residues in their epitope that are mutationally tolerant. And the mutations that accrued in these escape mutants are at these mutationally tolerant positions, and they're fixing mutations that we annotated as being more or less neutral. And so I think they really do highlight kind of how we can better focus on constrained positions, which might limit the potential for virus to kind of escape from population immunity if that's something that's possible in its future, kind of the way that we see with influenza.
Yeah, and I'm curious what you think about this, Vincent, since you've been kind of keeping a close eye on some of the vaccine development. Do you think there is enough diverse approaches right now that some of the variability like this is being taken into account, or what are the prospects that will be a few months down the road here and in good shape?
Well, if you look at the history of coronaviruses, there's not a lot of antigenic variation of that sort that would evade a polyclonal response. I think a monoclonal treatment, yeah, sure. If you just use one antibody, you could definitely get escape, and that's why in the Regeneron approach is to use 2. I think they're very closely aligned so that you have a lesser chance of getting resistance. But I think the vaccines themselves are not going to be a problem. I'm not worried about that. You also put some of these changes into pseudotyped viruses, which I think is great because it's just, isn't it great we have pseudotypes because you don't have to do a BSL-3 experiment.
Right.
So I'm really interested in the changes you put in that increased ACE2 affinity. You said they increased pseudovirus entry. What do you guys think about that?
I was actually shocked when to get that result. I really did not expect increased ACE2 affinity to result in increased entry with the pseudovirus. But I think perhaps that shows that in our pseudotype system, kind of the limiting factor, because really all we're measuring with the pseudotype virus is entry. Yeah, we're not doing multi-cycle viral replication. And the only gene from SARS-CoV-2 that we're using at all is spike, and everything else is lentiviral, backbone. So That's kind of the limiting step. And if we increase affinity, we can mediate more entry in this system.
But we definitely think, and Allie was saying this, and we spent a lot of time in the paper agonizing about trying to say this clearly enough. We don't think that these mutations that enhance ACE2 affinity are, we can't say for sure, maybe they would, but we don't think they're likely to increase the virus's fitness in humans. Yeah. The reason being that there's a decent number of those mutations there. They're accessible to the virus, and it hasn't fixed them so far. And I think this comes back exactly to what Allie was saying. The pseudovirus system measures how well the pseudotype lentivirus can enter a cell. But an actual virus infecting a human has to do a lot of things, including getting out of cells and infecting the right types of cells and all sorts of things like that. And in general, a virus can infect a cell. A virus needs to be able to bind to receptor to infect a cell. That's a prerequisite. But there's a point of diminishing returns, and binding to this receptor tighter and tighter isn't always better for the virus. So we think that the fact that they increase infection in the pseudovirus is at least possibly and probably likely something that's an artifact of the pseudovirus system rather than a reflection of the real fitness of the virus. Although until someone were to test that, we can't say for sure.
Yeah, and I think another important thing to point out with our pseudovirus system is that we're infecting HEC293T cells that stably overexpress ACE2. Yeah. So we put as much ACE2 as we possibly can on the surface and then infect them with our pseudovirus. So it is a very artificial system.
I think this is really important because, as you know, with D416G, many people are doing cell culture in HEC cells and making broad conclusions. And unless you're going to do a transmission experiment in an animal model, I'm not sure that you can say that. And you're making the right points there, right? You are really limited and Nels, you and I have talked about this before, right?
That's right. So obviously that D416G mutation has really gained a lot of attention and hype, I would say maybe is the way to say it. But I'm curious though, did that come through in your screen at all?
So D614G actually isn't in the RBD. I think the hypothesis mechanism is that it has something to do with increasing the stability, perhaps preventing premature proteolytic activation, although I haven't read those papers in.
Or shedding of the cleaved part of the spike,
Gotcha.
We did kind of pull out all of the mutations that have been sequenced within any of these RBD isolates or any of the coronavirus isolates to date. And in general, like mutations in the RBD are quite rare. None of the mutations we found in these sequence sets were present in more than like 0.5% of global isolates. But beyond that, we have, these biochemical phenotypes of these mutations. And so we were quite happy to see that kind of across the board, they're pretty much neutral with respect to these two phenotypes. And so hopefully that means we won't be falling into this kind of media hype of big functional effects in not real systems.
Yeah. I don't know, the virus is working pretty well as it is,
Yeah, exactly.
I don't know what the selection
very high affinity already.
I don't know what the selection would be, right? It's already transmitting really well. It's replicating to high titers and shedding and so forth. So
where we think to go to the evolution, where we think our data could be interesting is, clearly at some point before this virus first started to transmit in humans, it evolved to bind ACE2 better.
Yeah, sure.
And probably some of the mutations we find here are relevant for that. Yeah. But actually, as you guys have pointed out many times, compared to SARS coronavirus 1, we don't have a lot of sort of near ancestors of this virus that are currently known. Maybe people identify them. And then our data could help maybe interpret what happened in that evolution.
I so hope so, because that's the key. But as you know, NIH took away one grant to do wildlife sampling in China to answer just that question. It's crazy.
Yeah, no, really crazy. And it's worth, for our listeners, if you haven't caught up with that episode with Peter Daszak on TWiEVO. That was a few episodes ago, right, Vincent?
Yeah. Well, they're going by so quickly,
That's right. But I've been a little distracted with other matters, but that was one I caught, and I think I would really highly recommend.
Great. He's just great.
Describe both the science and, unfortunately, a little bit of the politics here. It's really important, I think, to have that view of what's really going on in real time here, and how, in sort of a critical juncture, we're potentially squandering the use of our resources put into the places where we should. And so it's an important part of the conversation here too.
You guys have any desire to eventually put these in infectious virus and see what happens? Do you have a BSL-3 there at the Hutch?
There's not a BSL-3 at the hutch right now, and we're not going to try to do that ourselves. I mean, we have like kind of contacted a few people to mention it, but one of our goals was also in sort of trying to publish this data as soon as we can we don't even know who all the people doing these experiments are. So we hope that maybe some people who are looking at mutations in the context of real virus will just take the mutations and do it ourselves. But we feel like the pseudovirus system is something we're confident in, but we think there's probably a lot of other labs out there at this point that are way better at doing infection. Well, we can't do it at all, but even if we tried, even if we got the facilities, would be way better than us at doing actual infectious virus experiments. And it would be really interesting to see what were the effect of these ACE2 enhancing mutations in real infectious virus. Although I have to say, even if they were found to be beneficial in cell culture for real infectious virus, I would still be suspicious whether they're beneficial in nature, simply because they haven't appeared yet. So it's hard to explain why they wouldn't have fixed yet if they're beneficial.
Yeah, I totally agree. Even if you found something in an animal, I'm not sure how you can say anything about people, right? Because they're different. But what would be interesting also is to take the lower affinity changes and see if you move them through cells, if they regain higher affinity, right? Would be interesting to see that, and whether that's dependent on just having ACE2 or a certain kind of ACE2. Yeah, I just wish we didn't need to work on this in BSL-3. A lot of things you could do that would be really interesting.
Yeah, no, I agree. And sort of consistent, unfortunately, I can't remember the name of the reference right now. I think it's the Lang et al. 2020, but there is, for people who are interested in this idea of affinity and viral fitness, there's a long history of literature, this in the flu field. There was a very nice paper by Lang et al. It's from one of the groups in the Netherlands. It's on the OC43 coronavirus, and they actually are experimentally showing that there's some balance. And just like with flu, the virus needs to bind to receptor pretty well, but it actually gets worse if the virus binds to receptor better. I mean, that's somewhat different than SARS coronavirus 2, because 43 has a sugar receptor rather than a protein receptor, but it does sort of emphasize, and it's a really nice paper, it's Lang 2020, that this relationship between receptor affinity and fitness is complicated. And
Allie, you said you were doing neuraminidase, and then there's a balance between HA binding and neuraminidase activity, right? Because if the binding goes up, the HA, then the neuraminidase activity has to as well, right, to compensate.
Right. Exactly.
I don't know if that's what you were working on or not, but I seem to remember some papers from Jessie's lab dealing with that, right?
Yeah, we've had some, although probably not the best papers on HA.
I remember one where you turned neuraminidase into an attachment protein, right?
Yeah, so this is sort of an artificial thing that just came out. It turns out neuraminidase can be that protein. Yeah.
I think we did that on TWiEVO. Yeah, it's cool stuff.
Yeah, I like this virus.
So Nels, anything else we should probably think about wrapping up at some point, right?
Yeah. So maybe just a quick question. What's next for this work? Are you going to continue doing it, or what's going on in that front?
Yeah, we have so many ideas that we're kind of just spending all our time talking about that. So Tyler, do you want to go first, and you can kind of let us know some of the things you're thinking of?
Yeah, I'm hoping to get back to some more evolution stuff. So I've just started kind of looking more broadly at the substitutions that occur in the deeper history of these RBDs, and hopefully mapping that kind of, as we were alluding to, not only to where human ACE2 acquisition comes from, but also what are these other host species ACE2s? How are those affinities changing across the phylogeny of these viruses? And so I think there's a lot of questions there. Even as I dug into like the SARS-CoV-1 literature, I think there's actually a lot of questions about the evolution there that never ended up getting fully resolved. And so it seems like there's a lot of interesting evolution kind of over deeper time to dissect here.
And don't forget MERS.
Yeah.
Be interesting too, right?
Yeah. I haven't even gotten to those papers yet.
About you, Allie? What are you going to do?
Yeah. And then another area that I'm super interested in is kind of using this library of mutants that we have now to understand how these mutations in the RBD affect the ability of the immune system. So either antibodies or polyclonal serum to continue to bind to the RBD. And so, how would mutations affect immunity and the implications that may have for, vaccine development and things like that?
Very cool.
Are you both in the lab all the time, or do you take turns? How does it work?
It's complicated.
Okay.
I've been, so I've had some family extra complications where I can't always be in Seattle. So I kind of come up to Seattle for a couple heavy duty experimental stints. And then Allie's just got the slow burn of always juggling 19 things.
Got it. And Jessie, you stay home, right?
I actually often go into my office and sit in my office with the door closed. Or sometimes I'm working from home.
Yeah, that's where I am now.
Yeah.
Nels, you want to do some quick picks before we wrap it up?
Yeah, let's do that. So my pick of the week, I'm just scrolling through here, is a song selection. So we've talked a little bit on TWiEVO about, our Trampled by Turtles theme has gotten, I think, some positive attention and motivated a few really fun conversations with some of our listeners who've sent in some song picks. And I've got one today. It's from a really good friend, an old pal of mine from college, a fellow called Chris Castino. And he started a band way back in the day when we were in college called The Big Woo. It's this is a band out of Minnesota. Tyler and my old stomping ground. Mm-hmm. And I guess you'd call it a jam band. They've kind of, developed over the years. And Chris is now also part of a solo act. He just released an album, but this song is just an acoustic number that he did recently, and relating to current events. So, I think we might even say that we're kind of in the midst of two pandemics. So the one that obviously we've been talking about a lot with SARS-CoV-2, but also as we've recognized even in the last month or two with the really tragic death of George Floyd, the pandemic of systemic racism, which is long running, but now somehow kind of on the surface, and much more obviously in our conversations and so Chris addresses this with a song he calls "So Much Work to Be Done." And I kind of wanted to lift this up as sort of an optimistic idea about how we can think about what we might do in these days, and not just the days ahead, but the months and the years ahead. And there's a great lyric in here about the ivory tower. And so I think this connects actually through to us as academics. And certainly in my lab, we've been beginning discussions about how can we do better when it comes to diversity, when it comes to all of these issues that we're grappling with on many levels. And so anyway, I'm putting this forward just as a positive note, and I think an artistic and inspirational note. So check out, it's a Facebook link where he's posted this video. It's about a 4-minute song. And I think it really has connected for me and has been inspiring. So,
good.
Yeah. How about you, Vincent? What's your science pick?
I have this visualization of data by Flourish, which you might have seen, but global deaths due to various causes. And it's basically that suicide, malaria, malnutrition, homicide, all the way down. And COVID at the beginning of the year is at the bottom. And then you click start and you go through the months of 2020 and it slowly ticks, and it ends up in June at the top killer of this year. And it's stunning to watch this thing go, because the COVID goes a few hundred, a few thousand, 10, 20, 30, hundreds. It just surpasses everything else, which is beautiful animation by Flourish.
Yeah, I agree. Really.
You must have seen this, right?
I have, and I'm glad you're including this, because it's really a dramatic way of sort of putting into some perspective about what we're dealing with, how this continues to develop as the viral pandemic continues. And it's, excuse me, it's interesting that, at the beginning, I guess this is global deaths to various causes. Yeah. And the top one usually, unfortunately, is suicide.
I know, that's really unfortunate, right?
Correct. And, that's another aspect of this that we don't discuss much kind of on the science front, but is the mental health ramifications of the pandemic. And so, you see the virus in terms of global mortality outstripping that, but suicide is right in there too, and increasing. And so, in some sense, it's even more drastic or consequential than sometimes we think about. And so I think, this is a really effective way of visualizing it, and hopefully, keeping focus on ongoing, in some of the trend lines in a lot of the states, certainly more in the more rural areas. We're not out of the woods, to say the least.
Nope. It's not going anywhere soon.
That's right. So I wanted to ask any of our guests today, do you have any picks of the week that you might put out there? Something you've seen on the internet recently, or anything?
Don't have to, but we're just asking to be nice.
I can Nels asked us last night, and so I know you guys are struggling to bring some non-coronavirus papers into the mix. So I wanted to flag a recent preprint from Michelle Hayes in Harmeet Malik's lab on a really cool system that I saw at a conference first a few years ago, and then her and I have become conference and then now Fred Hutch friends, although she's moved on to a postdoc. But it's a really neat paper on a sort of selfish genetic element, which many of us have heard of. It's the yeast 2 micron plasmid, which is sort of a molecular biology tool. And that's all I ever knew it as. And she did this really cool project mapping how this is actually a parasite in natural yeast strains. And she kind of genetically mapped some natural isolates of yeast that have actual active exclusion mechanisms of getting rid of this sort of parasitic plasmid. And I remember just the first time I saw this thinking, oh man, this is so cool. It's something that's just a molecular biology tool. And yet there's always some evolution there. And so I think that's a really neat recent preprint in the evolution field to check out.
Yeah, thanks, Tyler. And a good reminder too, that there is other evolutionary biology going on. And I've got a backlog of like really spicy preprints and papers. We'll put the link to Michelle's I completely agree with this great, exciting study. I mean, I'm a little biased since I was part of Harmeet's lab, obviously. But there's so much more that's been happening too. And so we won't forget that. And we're still, well, someday we will return to evolutionary biology in addition to SARS-2. And so it's always good to be reminded of that as well.
Yeah, when I release a non-SARS-CoV-2 TWiEVO, the numbers just... It's like I'm trying to sneak them in to get those people who came to hear about the pandemic to learn some other virology, right? But
yeah, well, and Vincent, you made me a promise that when my spicy new coronavirus discovery project, we might. But I understand we're still very much in.
Is it published? It is, right?
Yeah, so it just came out in Current Biology.
All right, we should do that soon. We can do that soon. Yeah. It's time to. In fact, last Friday we talked a little bit about the Ebola outbreak in DRC. There are 2 separate outbreaks ongoing. So a lot of important stuff. And I wish people would stay and learn about viruses, but just want to get their fix and leave, I guess. There's going to be another pandemic, folks.
That's right.
Learn some virology. All right.
So we'll also next time get to where we're running way over, but next time we have some great listener letters.
Yeah.
And so
next time.
I'm really excited to dive back into that as well. But I think we'll probably call it for this episode.
TWiEVO 57, microbe.tv/TWiEVO. You can listen on your phone or tablet, any podcast player. Please subscribe so we know how many people are listening. If you like what we do, consider supporting us. Microbe.tv/contribute and questions and comments. TWiEVO at microbe.tv. Our guest today from the Fred Hutchinson Cancer Research Center, Jessie Bloom. Thanks for joining us, Jessie.
Thank you.
Allie Greeny, thank you.
Thank you.
And good luck. Jessie doesn't need luck, but you guys do. And Tyler Starr, thanks for joining us.
Thanks for having us.
Nels LD is at cellvolution.org, over on Twitter, L Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. And this is just really fun to kick around some science in the company of some great scientists. And, we're doing this all by Zoom these days, not just podcasting, but also most of our science. But I still have to say I leave these conversations more energized, more inspired. And so thank you, all three.
Yeah, talking is good. People ought to try it sometime. Conversations. I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, stay curious.